Welcome back. I hope everybody had a wonderful lunch. My name is Eneida Hatcher. I'm at the National Center for Biotechnology Information at NLM. So welcome to session 5. This is virus classification tools. We'll have 20-minute talks followed by 15 minutes Q&A. Our first speaker is Cornelius Roemer from University of Basel at Switzerland, and he is presenting Clade and lineage assignment with Nextclade.
I'm going to talk about, so we're now in the tools section. So I'm going to talk more about the tool that Nextclade that can be used to assign lineages broadly, not just for single viruses, but generally for all sorts of schemes. And so what Nextclade solves is this problem that someone has come up with a nomenclature, MPXV, SARS-CoV-2 Pango, disease X, we don't know what it's going to be. And you want to be able to very quickly find out which clade or lineage a sequence that you've just got from the lab belongs to. And you ideally also want to be able to do that without having to build a tree. So in the past, for many systems, you had to build a phylogenetic tree and then kind of dig into reference sequences that might be close. And that takes time, which many people don't have, and expertise. So Nextclade tries to address that. And the last point is that it's ideally, you don't want to have to create new software for a lineage scheme. So Pangolin had to create, a Python package to be able to assign new sequences to lineages. And that worked. It worked well, but it's not really scalable. And moving forward, you want to be able to do that quicker with less maintenance and just use something that's readily available. And in a sense, Nextclade tries to solve those issues. I'm going to show you quickly how it works. Many of you will have used Nextclade before, but just to make it clear how the problem is solved. From a user perspective, this is how it works. So we already provide Clade designation and assignment for quite a lot of pathogens. So one of the things you can do is you just drop your sequences now. You don't actually have to specify which pathogen you have, as long as it's supported. In this case, I'm uploading some example sequences of Mpox. And the Nextclade will basically blast your sequences against all the available datasets we have and identify the most suitable or a set of most suitable datasets. In this case, it identifies that Mpox virus all Clade. And then you run it, and everything runs in your browser. You don't need to install anything. It just very quickly analyzes your sequences. And, it's a bit too fast, I guess. And it very quickly calculates a bunch of metrics, a bunch of information, but most crucial for our session here, it's a bit slow, is the fact that you can get these lineages here assigned. And as we heard earlier, there's a hierarchy of clades for Mpox. We've got the clades 1 to A to B, and then the outbreak and the lineage. And Nextclade is able to deal with that whole hierarchy and give you exactly, where something belongs on that tree. It also shows a bunch of other things which are more of interest to the people who do the actual sequencing rather than maybe public health folks who are most interested in getting the lineage that something belongs to. And you can look at, we have to skip that maybe. And you can look at where the sequences you just uploaded fall on a tree. So that tree that comes with Nextclade, it's part of the data set. And it very quickly places the sequence you have on this reference tree. It's a bit like UShER, just in a miniature way, using a small tree. So we'll go back. That's too quick. Yeah, and so you can quickly see roughly where in the tree your sequences fall, which is very useful when you want to get context on your sequences. You can also export your sequences, but I guess because for time reasons, I'll just move on. Yeah, so it does support this hierarchical nomenclature, which means it's flexible. What if you have multiple nomenclature systems you want to apply because there's no consensus, you can also do that and assign multiple nomenclature systems. There are a couple of additional features that are nice, I think, for Nextclade. One is that no data gets uploaded to any server. All the sequence data remains on your computer, which is quite important for certain fields. And that's because all the calculation just happens in the browser. It's It's entirely private. You don't need to install anything. You don't need bioinformatics skills to run it. It's fast, which doesn't really matter for most viruses. For SARS, it's quite nice if you can just quickly get the assignments and look at 3,000 sequences in a go, which I do quite frequently. It also works with partial sequences. So you can upload a spike-only gene or spike-only sequence, and you still get fairly accurate calls of which lineage the sequence belongs to. We're now, recently with Nextclade 3, are also able to handle quite a lot of divergence. So most, you can align Ebola virus Sudan against Ebola virus Zaire and various dengue viruses against each other. So that now also works and broadens the applicability. And yeah, we have a high performance CLI version, so you can run it on all of your SARS-CoV-2 sequences, 60 million sequences in a couple of hours on a laptop, which is something that's been also an issue for some databases to run it quickly. And just to give you a brief idea of what Nextclade does, so the first step of Nextclade is to simply align the sequence you upload to a reference. There's no magic really there. We do it in sort of a quick and quick way, which works well. Math might give you a better alignment in the end, but it's good enough for the purpose we're looking at here. Then we extract the coding sequences from the aligned sequence, so we can translate, produce the genes that you're interested in, spike and so on. We can align those, so we get the the amino acid mutations precisely as they are. And we place on a reference tree to get the clade assignments or lineage assignments, and in the end present the results to the user. And I've underlined here the key config files that we use to Nextclade to a particular virus. Nextclade itself is agnostic to the exact virus you use. It's done in a way that you can just, it works for Mpox because you have some config files that specified that make it work for Mpox. If you switch out the reference and the annotation and the reference tree, it works for SARS-CoV-2. So those are really the three types of key files you need to provide for it to work with a new virus. And we call this sort of set of config files a data set. And these data sets, we host them on GitHub because it's easy to automate things on GitHub. It's reproducible. People can then just look at the code. They can also update suggestions, fix issues. And something that's another key design decision is that we want to make it easy for people to create new data sets on their own. So there's no requirement for Nextclade developers to approve your data set. You can just create your new data set and use Nextclade with it as is. There is this repository which you can submit your data set to, and then it automatically appears in the default Nextclade web page, right? But in principle, you don't rely on that. And in fact, one of Travis's students just made a data set for PRRS for a swine virus, and that just worked. And we found out because we saw that there was traffic from Iowa State URL, and later on we then kind of invited him to submit his data set to the index, and now it's available there. So now if you want to analyze PRRS sequences, you can just upload the sequences, and they will automatically you will get that data set suggested as a data set that fits the sequences. And Michael, he was just able to work and make this data set basically off the documentation with a little bit of help of some Nextclade folks. But that's what we're trying to encourage. We can't possibly maintain, data sets for all the thousands of viruses there are. It's very much the best thing if people make data sets and then kind of deal with the nomenclature who really study those viruses in detail. So we're trying to encourage that, and there's a tutorial available. Maybe I don't have to go in too much detail here, but essentially boils down to the three things, which is you just pick a reference sequence. Usually you can just use NCBI refseq for that. You have to sort of tell Nextclade which genes you want to have translated. You need to make a reference tree, which can be quite simple. It doesn't need to have many sequences. It's a standard Nextclade tree, which many people been very good at making themselves. You put it in a folder, and if you want to have it listed on the standard Nextclade tool, you just submit a pull request, and it becomes available for everyone. So if you have any questions, feel free to reach out to me, and we'll try to make it work. I don't know how much time I've got left. Time. Oh, 1 minute. I should have had a clock myself, but 5 more minutes. Excellent. So that was the part on Nextclade. I've got a few more thoughts that kind of came up because I was listening to the talks earlier today and yesterday. And I've been involved with sort of the two schemes to classify SARS-CoV-2 phylogenies. And there are the coarse next-strain clades, which try to capture things at quite a broad level. And we use these year letter clades to make it very quick to identify sort of when something is from. And they're very useful for high-level reporting. The other thing are the fine pango lineages, which there are more than 4,000 now. There are, in comparison, only about 30, 35 next-strain clades. But they sort of, each clade schema has certain advantages and disadvantages, and I've got a couple of illustrations to show some of them. So, for example, if you just want to understand at a broad, high level, the evolution of SARS-CoV-2 over time, it's quite good to have something that's a little bit more schematic or has fine-grained, is quite broad, like next-strain clades. You're not supposed to look at this. You don't need to read what's here, but you can just see the scale. There's just 40 lineages, or clades, and you can very quickly see what descends from what. On the other hand, on the other side, you have those 4,000 pango lineages, and every dot here on that tree is a pango lineage, and it misses some of the early ones, but it gives you an idea of the kind of different scales you see here. And well, each is good for different purposes. For example, covariance makes these Muller plots, and it's quite useful to have things at this very broad next-strain clade level. Otherwise, it would just be a mess. There would be 4,000 different lines in there, and you wouldn't really see anything. Whereas here, you can see that, there was BA1 and in purple, there was BA2 and was lighter shade of purple, and so on. On the other hand, one advantage of having lots of lineages is that people can actually use them in papers as soon as they are available. For example, here BR2 or BA2320, they are not part of, they are part of the BA2 or BA275 next-strain clades. We call them, I don't even know what the names are, essentially, because they're just colors, but people can use those lineage names in papers and therefore communicate very effectively which exact lineage they've studied. And you can query, for example, be it BR2 or BA2320 on Google Scholar, and you find all the neutralization studies. Whereas if there was no name for it, it would be very difficult to quickly aggregate all the available results. So that's one thing. Also, another argument for sort of a more fine-scaled schema is you see here, Trevor Bedford and team, they've got a essentially nowcasting tool for SARS-CoV-2 lineages in various countries. And on the left-hand side, they do that on a clade basis. And you see that, well, it looks like there's not much going on. You've got the BA286 clade, going to near dominance, and then it appears that nothing is happening. Whereas if you look at, on a more fine-grained level, pango lineages, you see that actually as part of those BA286, there is quite some evolution going on, and you see that one lineage here appears to kind of rise quite frequently, and whereas other lineages disappear. So in that sense, it's useful to have these different granularities to be able to do modeling, prediction, and talk about it. Some further thoughts, if I have time, are that I think something that Pango did really well was to put the ground truth of the classification system on GitHub. It's kind of unambiguous, it's transparent, people can see what it is, they can talk about it without having to go and look up in some paper. And it also enables independent tool development. So, for example, Nextclade, I got involved basically through the community, through the GitHub repository. I got involved in the Pango work because it was open. Had it been sort of some committee that works, without having this very public way of getting involved, I might not have actually, gotten involved there. And Nextclade supported Pango lineages at some point, and it was possible to do that so easily because all of the definitions were live on the webs on GitHub available, and I could just download from there and work off that. So that's maybe a pattern that's good to consider in the future for other viruses to really put out the ground truth there. And it also prevents you from having those issues with, what do we do when the funding runs out, and maybe the website is no longer able to function. If you have something that's on GitHub, it's very portable, and someone can just carry on the work, fork it, put it on GitLab or whatnot. Yeah, so that's just again illustration. We've seen them earlier. These issue boards, they work as a way to get people involved for Mpox as well as for SARS-CoV-2. There've been, I remember, I think for Mpox we got some public health folks from South America, opening an issue and being like, we found this cluster. Would it be possible to give this a name? And they, brought this to the attention of the people who designate those lineages, and we designated it, and it might help them tease apart the transmission chains of Mpox in that country. So yeah, that is pretty much it. I think to close off, I think the workload has come up with maintenance a bit, and I think I sort of do this designation as a side effect of studying the virus. It doesn't take up that much time to do it, and sort of if I kind of lost interest, then it would maybe mean that there's less granular lineages, but that in itself isn't necessarily a problem. I get sort of people complaining that there are too many lineages, other people want more. So there's no best way of doing it anyways. So I think, and for Mpox, it works really well as well. So I don't see that as kind of a very big issue, as long as there's good tooling to make it easy to designate. So that those are some things I did quite early on when I started designating. I just tried to make it very easy to, a couple of seconds to designate something new. And in the end, Nextclade is the work of Richard's kind of brainchild, and Ivan Axamento is a fantastic developer who's been doing a lot of the great computational work to put that in practice. Yeah, thank you very much.
Next, we'd like to welcome Angie Hinrichs from University of California, Santa Cruz. She'll be discussing UShER and autolin, identifying virus lineages.
Well, thank you to the organizers for inviting me to speak on behalf of UShER and autolin and how they can be useful for finding new lineages. All right. So first I'll introduce our tool UShER. It stands for Ultra-Fast Sample Placement in Existing Tree. I'll briefly show off our big tree of 16 million plus SARS-CoV-2 genomes that servers at UCSC are crunching away on updating as I speak. I'll talk about a couple different roles that UShER plays in the Pango lineage ecosystem as I see it today. And I'll delve into our tool autolin, which is an attempt to automate some of the work of finding new lineages. But first, I'd like to highlight some important differences in phylogenetics for non-pandemic viruses versus, exploding pandemic viruses. So for many human pathogens, we might have, a few thousand sequences in GenBank that have been sampled across decades. Whereas in the SARS-CoV-2 pandemic in 2021 and 2022, when the testing and sequencing had really ramped up, we had millions of sequences, and we're getting tens of thousands of new sequences in the repositories every day. The genomes from these pathogens that have been around forever tend to be very highly diverged, whereas in SARS-CoV-2, the sequences are relatively very similar to each other, just lots of them. For a traditional pathogen virus, since they're so highly diverged, you really want to use the maximum likelihood estimation algorithm when building your tree, because for highly diverged genomes, it builds better trees than simpler approaches like maximum parsimony. However, at pandemic sequence scales, maximum likelihood estimation would be too slow to do that way. So you need new methods. So in response to all these challenges of scaling up, our group at UC Santa Cruz, led by Professor Russ Corbett-Dettig, developed UShER as part of what we like to call an online phylogenetics approach. And here online is kind of in the classical computer science sense of incrementally updating a result when you get new information instead of recomputing everything from scratch when you get new information. And this is what enables us to scale to high volumes of data, just building on the tree we already have when we get new sequences. So long story short, in 2021, we started using UShER to add new sequences every day to the last public release of Rob Lanfear's SARS-CoV-2 phyllo tree. And today it's over 16 million genomes. Here it's shown in Theo Sanderson's taxonomy viewer, colored by Pango lineage. And you have to zoom out really far to cram 16 million sequences into some kind of representation. Now, the full tree of 16 million sequences, I can't just post on the internet because it includes GAZAID sequences, which have restrictions on sharing. But there is a tree about half the size of the full tree that has only the GenBank INSDC sequences and also COG UK direct release and the GenBase data from China. So those public sequences form a tree of about 8 million. And Theo Sanderson picks that up and puts it on cov2tree.org. So to make the full tree as accessible as possible, we added a web interface for UShER. You can type usher.bio into your browser. And then if you have SARS-CoV-2 genome sequences, you can upload them. Then we'll run UShER on our servers and place your sequences in that big tree, and then extract subtrees, which are kind of the branches where your sequences land. And we'll give you the subtrees so you can see the most closely related sequences and what makes them different from your sequence. And the best thing about it is it links out to Nextstrain for viewing. So you can view these subtrees interactively in Nextstrain's great viewer. And now we also link out to CDC's microbe trace tool. So this is kind of my perspective on the Pango lineage ecosystem. You've already heard its history from Anya, but briefly to recap, this is how it is, as I see it today. Of course, it all starts with scientists all over the world sequencing SARS-CoV-2 genomes and critically sharing their genome sequences in data repositories. So every day, UCSC, we pull the new sequences from the data repositories and add them onto our tree and run some optimization to make sure that we have the best placements possible given all the current information. Our web interface and the tree are used by this volunteer team of lineage hunters, saltation spotters, whatever you'd like to call them. These are people who just have taken this up as a hobby, no matter what their profession is. Now they spend time looking at all the new sequences every day and placing them on the tree, and they're looking for branches of the tree that are growing, and most often they're looking for spike mutations that would be especially concerning. When they think they have found a branch that is worthy of being a Pango lineage, they'll propose it, and then a lineage designator, who at this point is just Cornelius, will evaluate those proposals. And if they meet the loosely defined Pango criteria, then Cornelius will add that new lineage to files in this GitHub repository, Pango designation. And I want to reiterate what Cornelius said about what it means to have this process open and on GitHub. That's how I got involved too, just by commenting on issues in this repository when I was starting to build the UShER tree. Like, well, the UShER tree has this to say about that proposed lineage. And Anya kind of welcomed us in. And so, yeah, that's how, it's been sustained so far. So Pango designation in turn informs the pangolin tool that assigns lineages to all the new genome sequences. So I see it as, kind of a closed feedback loop. These lineage assignments are going back to all the scientists who share their genomes. Another little feedback in the system is that all the new designated lineages need to be annotated on our UShER tree. That's how we get the pretty coloring by Pango lineages. So every time Cornelius designates a new lineage or 24 new lineages, I need to pick out the right node of the tree and put a label on that node for the lineage that gets propagated from one day to the next as sequences are added. I also get a lot of feedback about the tree from branches that look weird to the lineage hunters. So that's another kind of service they provide. Really is quality control. They might find a branch that looks weird because maybe amplicon dropout is causing, like as Betty described earlier, false reference values that can really cause trouble in a tree. So I might mask positions in the genome that are particularly prone to sequencing errors like that on a certain branch, or exclude some sequences that'll just look like they came from a bad batch. But that's the ongoing maintenance work for this tree. Now a minimized kind of severely pruned version of the big tree goes into pangolin and is part of how pangolin assigns lineages. It's running UShER internally, and the placement on the little tree is where these lineage assignments come from now. And I'd like to, as Anya and Cornelius already have, point out that so much labor is coming from volunteers. And on the one hand, it's a beautiful story of open science and citizen science. But on the other, it's rather important, isn't it? Do we really want to just leave it to, volunteers? Also, SARS-CoV-2 grabbed the attention of everybody in the world. So it can attract dedicated volunteers like this. But there are so many pathogens that, hopefully someday we'll have more sequences for more surveillance. And, can we just hope that volunteers show up to scan for new lineages? I don't know. But, I hope that some funding body will fund a home for lineage maintenance. But that's kind of above my pay grade. From the technical side, though, what can we do? We can try to automate as much of this work as possible. So what are the lineage hunters looking for when they're scanning the tree and searching for new sequences every day? They want to see distinct branches that have some kind of epidemiological significance. These are the same epidemiological events you saw in Rachel's slide earlier. And here I've colored them by how well they lend themselves to being automated. When you have a tree and basic metadata, such as the date of sample collection and the country of sampling, we can automate the search for branches that are growing. We can automate searching for introductions into new geographic regions. We can highlight specific mutations, like in the spike protein, that are, considered more likely to have a phenotypic effect. Now, recombination always makes things harder when you're using a tree. It's not really the right data structure. We've got some ideas about how to deal with that, but that's more future work. And then wouldn't it be nice if we could automate the search for change in phenotype, but Who knows really how to truly automate that? And even if we wanted to search in metadata, the metadata that we might need to do that is not always available. But, so we'll just focus on the first three. The program that's attempting to do all this was written by Jacob McBroom as a graduate student in Russ's group. So as its input, it takes a mutation annotated tree, and that can either be the UShER tree of all the SARS-CoV-2 genomes, or it can just be a Nextclade build. And it has a way to pick out genetically distinct branches that we think are pretty comparable to what a human might pick out just looking at the tree. And the number of branches that it could pick out can be a little overwhelming, since you still need a human to evaluate these and see, like, do they really fit the criteria for a new lineage? So it has a way to prioritize all these possible new lineages by their growth rate and by a very flexible and configurable weighting scheme that I'll talk about in a minute. But it can either come up with a new lineage system from scratch that's kind of Pango-like, or it can take an existing system like Pango and then propose extensions to it. And importantly, it scales to pandemic flood volumes of data. So first, how do we find lineage-looking branches? For this, Jacob went all the way back to information theory and framed the problem as one of communicating about genotypes using lineage symbols as a system. So the manuscript has this nice long mathematical derivation of this genotype representation index, but this is what it ends up looking like. A simple function that can be computed rapidly for every node in the tree. That's what makes it scalable. But the basic idea of this is that if you have a branch of the tree, like the one on the left, that has many descendants and kind of a reasonable amount of divergence within that branch, it ends up with a higher score. However, if you have a branch like the one on the right, where it doesn't have so many descendants and there are long branches, long distances between all of them, that gets a lower score. And, kind of anecdotally, what we've observed for SARS-CoV-2 is that when you have a branch like the one on the right, it's usually a bad sequencing batch. And a lot of sequences. So we like the branches that look like the ones on the left. But now it can pick out an overwhelming number of those branches. So here is a snapshot of the same branch on the tree. On the left, you can see that human curators have picked out 4 of those branches to be new Pango sublineages. On the right, you can see a lot more different colors. That means autolin has picked out pretty much every branch in this neighborhood as genetically distinct. So we really need to rank those. So the first way is to rank them by how fast they appear to be growing in recent weeks. Basically, we fit an exponential growth curve to data that's normalized by the percent of sequences on that branch from each country. If the curve fits well, the upper and lower bounds of the confidence interval are not too far apart. If the lineage is not really growing, then you will get kind of a poor fit for that curve. And in either case, using the lower bound gives you an estimate of like kind of both the growth and your confidence in that growth, kind of a conservative lower bound approach. So that's how we rank by growth. And then we also need a way to weigh some samples more than others, as has already been brought up multiple times. Sampling definitely is like overrepresented in certain countries. Some countries sequenced vastly more genomes than others. So in order to avoid that, biasing our ranking of lineages, we can add weight to samples from countries that did less sequencing. And And finally, some mutations are known to be more interesting than others. So you can have a completely user-defined system. For example, for SARS-CoV-2, Jacob has a script that pulls all of the antibody escape scores from Jesse Bloom Labs' online calculator and converts those to weights for autolin. So these concerning mutations can be flagged and bump that branch up to the top of the list for evaluation. And there's a web app, autolin.bio. You can drag, drop in a next strain build. There's a link, to the chikungunya one that you can download and try it out. So wrapping up, we've found that this approach of maintaining a big daily updated tree and making it as accessible as possible has scaled with the volumes of data. It's been somewhat useful. And it's been important to us also to, make it so that anybody with internet access in a SARS-CoV-2 genome can use our resource to get placement in this 16 million sequence tree for their sequence. So final conclusions. We have now great tools for going from genome sequences to lineages. And I am excited about Nextclade as a possibility for all the viruses going forward, because Cornelius and Ivan have really architected it to be extensible to many pathogens. But these things require material support. We need to maintain, like all the genome sequencing that happened for the pandemic. We need to increase that sequencing across the world if we can. And lineage system maintenance, so far that's been largely a volunteer effort for SARS-CoV-2, but it's something that requires support. And in Celeste's talk, we saw how that can go away, for example, for rotavirus. So any way you can support the maintenance of these systems will help a lot.
Thank you. And finally, we have Christian Zmasek from J. Craig Venter Institute in BV-BRC, and he'll be discussing BV-BRC subspecies classification tools.
Thanks to the organizer for inviting me, and thanks for people for listening. So basically I'm going to talk about the subspecies classification as it's currently implemented in BV-BRC. But actually my main point of this talk is not to advertise it or to explain it in great detail. It's more like to get input on further extension and how I can make it better, and both make it better on the algorithm side or the computational approach, but then also because our approach relies on it relies on reference trees, which are of course in general generated by the experts on any particular virus family, and so I'm not an expert on any family, so we need the input and in a kind of way the approval of the experts on any particular, like hepatitis C virus or rotavirus, so to improve our tool and make them generally accepted by the community. So now that's kind of the purpose of my presentation mostly. And so the subspecies classification tool in BV-BRC, you can find it under bvbrc.org under tools and services and subspecies classification. And it's relatively simple to use. You just depending on the virus you're analyzing, you have to paste in a sequence. It could either be a whole genome, or it could be just a particular protein sequence, and then the program will run and it will produce it gives you an assignment for a subspecies, which could be a clade or a genotype, depending on the virus family. And it also gives you an image of the reference tree with your query sequence placed. So that's how it works. And the computational background behind it is what we're using. We use, as I already mentioned, we use reference trees and reference alignments, and then we use a program called pplacer, which places the query sequence on every possible branch of the tree and evaluates the likelihood of that particular placement. So it's a maximum likelihood based method. And so currently, so those are the viruses we are currently covering. So, and also you can see what the query sequence is. For some of them, it requires it's just one. They're just based on one gene, and others are based on a whole genome. And it's all, as I already said, the tool is it's maximum likelihood placement. The exception to this is our tool for the rotavirus A, which is it's based on a indeed now outdated specific tool, and so that's of course is our goal is to replace this specialized tool with the maximum likelihood placed approach. But as you can see, one key difference is between rotavirus and all the others is that for the rotavirus community, when they assigning a subspecies, they use a subspecies for each segment. So that makes very complex subspecies, whereas the other ones, they are based on just either the whole genome or a certain segment, so you don't have this issue. So the computational approach for rotavirus, so maybe other viruses like it is quite different than the others. So, and we are aware of that, and we are currently in the process of expanding this. And so, as I said, we already have, so these viruses are covered, and even here, I know I kind of went through the reference trees, and I noticed some of them actually are outdated, so they need to be updated. But so that's the situation here. And then we started, but basically as an intellectual exercise for the preparation for this workshop, we started to add additional viral species, and for now we kind of focus on adenovirus, so we added a couple of those. And then also, due to current unfortunate events, also we started working on measles and mumps virus. And so those trees they're tested and they're ready to, they're already in alpha version, I think. And currently we're working on the RSV virus, which has a little bit more, its subspecies are a little more complex. They're also like hierarchical, like influenza, for example. So, but also more interesting, I would say. Whereas some of these flaviviruses, the subspecies, at least the one I'm aware of, they're relatively simple. They're just like, there's no hierarchy. It's just like A, B, C, or 1, 2, 3, 4, 5, 6, 7, something like that. So that's kind of current work. And here I kind of hope that as a result from these workshops that actually I contacted or I'm contacted by the experts on those particular viruses, so that we can make sure that the reference tree we are using is actually acceptable by the community and, reflects the current status of the work. So that's I said that's kind of one hope for me for this workshop. And just one more time, so that's kind of how our pipeline works. So we have hopefully an expert approved reference tree, which has to be calculated by RexML, and also with the corresponding multiple sequence alignment. And it produces a statistics file. And then we have the user query sequence, and then we use MOST to align the query sequence to the reference multiple sequence alignment. And then we use the pplacer program, which is in the reading material if you want to learn more about it. And then use another program called Copy to which is is needed to process the output from pplacer. And then here comes the Clade. That's the program developed by us. So that's the program which actually takes the results from pplacer, which can be quite complex, and kind of summarizes them and makes them more easily readable for people, and also produces a machine readable output. And then this goes into the BV infrastructure, which then pplacer is also a user, which is as I said, it's a user readable output, and also a phylogenetic tree where you can see your query sequence. That's more like in generally, we hope that the textual output is so good that people don't need to look at the tree, but still many people, they want to confirm the result by actually looking at the tree. So that's provided too. So that's how it works. And the rest of the second half of the talk, or second to third, I'm going to talk about the Clade program. As I said, that's the program which is being used for to analyze the placements in hierarchically annotated phylogenetic trees. And here's my hope. I'm currently working on version 3, and it's always dependent on the user community. I kind of change the program in various ways because people have kind of, have different wishes how they want the results to be presented to them. And I hope that, as I said, that's my second hope, is that as a result from this workshop, that I can get useful input what people really want in the output. And the main, probably most people here are familiar with the concept of using reference tree and placing of sequences on the reference tree to annotate those sequences. So in most cases, it's easy because if it falls into a clade, your query, it's clear it's a member of that clade. But I thought the more interesting cases are the ones where it actually falls outside of any kind of clade. And that can mean two things. It could mean the program is wrong, but it also could mean that actually have a first representative of a novel sequence, which is in generally, or not a novel sequence, a novel subtype, which is useful. Or it could also mean that an existing clade or subspecies needs to be expanded to, actually that particular subtree is bigger than as previously thought. So, and I thought that was there's the third case. It could be that when you get a weird placement, it could also mean that your query sequence is in some ways it's corrupt. Maybe it's too short, or it's not even, maybe it's a human sequence or something like that. So it shouldn't even be there. So, but anyway, so that's kind of that's the easiest case where we have the query sequence and it falls, inside in this subtree, which I label with A. So it's very clear. And I usually use the term bracketed, so that means bracketed by A's, and it means it's of A type. That's the most easy case, and that's very intuitive. So, but then, as I said, to me that's an interesting case where your query sequence actually falls. It doesn't fall into any existing clade. It doesn't fall into the A clade. It doesn't fall into the B clade. It's kind of in between. This mouse is really slow. So it could mean that maybe A is actually bigger. So maybe that's simply Q is actually, it's also an A. It could mean it's a subtype of A, which may be called AX, A.X, or it could be completely a new subtype C. So we don't know at this point. But so that's something that the user should be warned on if that happens. We shouldn't just say it's A, because some people might, it's kind of the naive assumption it's just another A, but it's not necessarily true. And then we have here this kind of case where our query falls between like A.1 and A.2, and that means we can be sure it's A, but again, we don't know. It could be A.3, or it could be A.1.1, something, or A.1.x. So that's kind of another situation. And so, as I already said, for this purpose, I wrote this program called Clade. It's a command line program. It's not intended that the user run it. It runs behind the scene, and it produces usually two outputs. One is human readable, and one is easily parsable. And it's, as I said, currently I'm working on version 3, and the next couple of slides, those are what I'm proposing for version 3, how the output should look. But as I said, I'm just one person, and that's what I like, but maybe community likes something else. So that's why I'm presenting it, so that maybe I can get input how to make it more, whatever people want to do. So another good thing about the P-Placer program is that actually it's likelihood based. So it can actually give you multiple placement if there's uncertainty about the placement, which in this kind of case is not here. We just have one placement. And so here's the proposed result. So the consensus is this one is A1. That's very clear. It must be A1. And it's bracketed by A11 and A12. And probability is 1 because there's only one placement. And it could be a potential new A1 subspecies because it's between 1, 1, and 1, 2. So that's kind of part of the output. And then here we have another situation here. Again, consensus is A, and it's bracketed by A1, but then the rest of A is not clear, or it's bracketed by A2, and there's a uncertainty behind that. Again, probability is 1. It's potential for novel subspecies. And then here we have, now that's interesting. So we have like multiple placement, which especially if it's like that, if it's like split evenly, that probably means your sequences might not be good. It might be too short or something like that. But nevertheless, consensus is A. It's bracketed is not really available because it's not bracketed by anything. Probability of placement within A is still 1, but there's an uncertainty of placement within A. So, and now we have here a case where we have like 3 placements, which is very similar to the case before. And I need to check the time if I'm not over. Okay, still good. So here we have A. Probably it's A. Sorry. Oh, it says don't use a mouse. Okay. So here we have placement within A with a probability of 0.9 because there's a small chance it might actually be outside of it. And consensus is A, nothing bracketed, uncertain placement within A. So that's also pretty easy. Then we have a very similar case. We could have the probabilities are now a little bit shaky here. It's 0.7, but still probably A. And then here is a case where that probably means that the query sequence is, it's a good chance it's not, it's something, it's too short or it's wrong, it's a human sequence. So we have, there's no consensus and uncertain placement. So here we cannot really say anything, what it might be. Probably should give a warning to the user, please recheck your sequence. And here's another case where we have, there's no consensus because it's between A and B. It's bracketed by A1 and B1 with a probability of 1. And this was a high potential for a novel subspecies. So that's extremely likely that's something new, and that should be very interesting, and it should be flagged when it, or should be, it will be in the ascending version 3, it will be flagged to the user as such. And then here we have a case, we also have no consensus. It's falling out of A, I mean, between A and B. Again, this one will have very likely a novel subspecies. So, and now that's just repeating that. So that's how our pipeline works. We have maximum likelihood placement based on mostly math and p-placer. And the program which summarizes the result is called Clade, and that is being used within the BV-BRC infrastructure. And that's it. Thanks for listening.
Okay, so we will start 15 minutes of question and answers. Is there anybody in the room who'd like to go first? Okay. I think I saw Richard first.
Hi, Richard Shorman. For Angie, so as you're starting to add these sequences using UShER, like at some point, it seems like if you rebuild the tree, you would have a different structure than if you're incrementally adding in. So how do you determine like when it's time to reconstruct the reference tree?
Richard, it hasn't crossed our minds to do that. I'm just kidding. It would take a while now that we're up to 16 million sequences. Also, having seen some other attempts to build trees from scratch at this scale, the cumulative curation effort that has gone into the tree over time as people and, these lineage hunting volunteers find little errors here and there, and we find, oh, we better filter out these sequences, oh, we better mask this position. When someone attempts to build a tree from scratch, if that same level of care isn't taken, you end up with really weird branches being possible because of sequencing errors. So, the UShER tree is not the ground truth. It is not the one true tree, the only one or best one that's possible. It's just a tree that works well enough.
just a further comment on that, when I started looking at recurrence of mutations, I was so grateful for the strategy that you have, because by putting the new branch into the older branches, you preserve the time structure. So you can, it's not going to be perfect, but I think it's closer to accurate than if you rebuild, because there's so few changes between the different clades. So I'm really happy that that's the method that's chosen, not only for the good reasons you describe it, for that other point as well. It's beautiful that way. And I had another comment for the last talk. I was thinking your in-between clades could be recombination if the virus supports recombination. Those could either be new clade or recombinant.
That is true. That should also be.
And that could be something you could automate and place into the program. And even if the virus doesn't support recombination, you can have it in the test tube. So, it might be a good sequence to flag and look at from that point
That is correct.
of view.
I kind of forgot to mention. It's also, that's another.
Yeah, it's another real possibility.
Yes.
Thank you.
Thanks for the comment. Yeah.
Yeah, this is Richard here. I have one question, maybe for kind of all of you. So in the last talk, we've seen this issue. How do you deal with a placement that's kind of in between, right? But in all your examples, every lineage or whatever group was the single tip, right? Which might have been an abstraction on your slides. But in order to kind of answer the situation or give an answer in the situation that you discussed here, you kind of need some sense of a diameter of these individual groups, right? And for SARS, it's kind of 1, 2, 3 mutations, right? But for other things, you kind of always need 2, 3, 4, 5 sequences to have some sense of how what the diameter is. And I think then you can sort of meaningfully start to give an answer to the questions that you posed.
I mean, for my slides, yes, that was my intention when I had like one tick. That didn't mean just one sequence. That was supposed to be like a whole subtree, like an A subtree. Not just yes, you should always have. In my estimation, should usually have at least 4 sequences per sublineage or sublineage. So that's that.
But yeah, then if something is like one mutation outside of your subtree, you probably want to lump it into, right? And if it's kind of halfway in between 2, then you need a new name.
I mean, that's I kind of forgot. I forgot to mention during my presentation. What I have left out is the issue of branch lengths. I mean, that adds another dimension, which is, and I would kind of like to discuss this one with like individual people working on particular trees, because the branch lengths are so very different for different virus families. But yes, there should also be a cutoff based on a branch length. If something really close, even if it's outside, but it's extremely close, it's probably still a member of that. But how close? That's for me. I cannot answer this question. So that's why I personally prefer only work on the topology. But most people, they also like to incorporate branch lengths and like the measure of closeness, how close something is to any other subtree. So that's definitely another complication. So yeah, I personally
addressing that kind of from the SARS-CoV-2 UShER Pango side, in the Pango lineage system, everything is defined back to the root of the tree. And so, placement in the tree, if it's not A.1 or A.2, it will just default to A. And we have every node at least has an ancestor that is labeled as a clade. So it there's kind of a default. And that's the UShER web interface just shows you the top placement. So we are not doing what you do with showing the multiple placements. So that's definitely a area of improvement.
We do have the problem sometimes that you get extra sequences that fall in between. Say we define BA286, and now something pops up and it's kind of halfway between BA2 and BA286, and then the question arises: does it belong? Does it broaden BA286, or is it BA287 or something like that? And we kind of try. It's you who kind of has to make a decision, because you annotate exactly on the full tree where it belongs, whereas Nextclade just kind of reconstructs a founder sequence and then does the nearest distance based kind of nearest neighbor matching. But I do help a little bit sometimes by annotating mutations that I think should belong to something And I think it works quite well. I remember you sometimes ask, where should I draw the line when it wasn't so clear? Luckily, it doesn't happen very often. But having sort of this, I think at MPOX, we do it redundantly. We have reference sequences and we annotate mutations that are defining, kind of, because that gives you some resilience.
Yes, from the tree perspective, I do need a set of mutations that are defining. I kind of winced when Cornelius talked about sequences that are sort of halfway to a variant, because often that's amplicon dropout. We get so many sequences that look intermediate, but from their sampling date, it's clear that no, they're just basically partial with the reference filled in different ways, and so we end up with a lot of mess around the base of each of these major variants that some pruning is sometimes required.
That's
Related to
That's actually, since I'm the speaker, I'm like, maybe I'm not supposed to ask questions, but that was actually my question for the Nextclade. So that's how it works. So basically, it works based on similarity to reconstructed ancestral sequences.
Just take the Pango lineages and infer what's kind of the likely founder. And it does have problems with things that are kind of right before what should be where the lineage starts. And Nextclade kind of calls that belonging to the lineage, even though I suppose UShER and being totally corrected with it shouldn't be part of the lineage because it lacks one thing, but because Nextclade approximates, it will call that to be part of it. And that's an imperfection we have that UShER doesn't have because it has a better resolution of it. But in practice, it doesn't.
Around it.
You have a hack, but it doesn't. In practice, it doesn't really cause issues because because usually when a variant of interest arrives, it grows fast, and so we usually don't find ancestors. Because in practice, usually things descend from the founder. There's very little that comes before, just because things grow exponentially when they.
The founder, you mean a real sequence, not a reconstructed sequence?
Well, reconstructed sequence. Oh, I see. Or what would have been the founder genotype based on ancestral.
So I actually end up setting additional lineage labels further back in the tree to catch these partial sequences, because I wouldn't want a BA.2.86 sequence to not be assigned BA.2.86 because it's missing a few mutations. So that's the hack around it.
Let's do an online question real quick.
The first question comes from Lakshmi from CVDC. Is Nextclade useful for virus sequence from livestock and poultry?
Yes, it could be applied to all sorts of viruses, even HIV. I think Richard has played with. Although there are difficulties with recombination, but the sort of is Nextclade is useful, but you need to make a data set, but it's not very complicated to make a data set to make it somewhat useful. There are kind of degrees of if you want a very complex lineage system like Pango with 4,000 distinct lineages, then it takes a little bit of care to make. But if all you want to do is call 5 genotypes in livestock in some virus, then that should be easy to do and should be useful. But I think that's kind of for the potential users to decide whether they find that informative. But it is technically possible to do.
Andrew at UVA. I was curious along the lines of Betty's observation about recombination, if there are subroutines in your different respective environments that handle automatic recognition of that, or is it just down to manual curation and observation? Thanks.
Go ahead.
Yes, at some point I'm planning. A long time, well, maybe not that long time ago. You might remember Viper, which is the predecessor to BV-BRC. So there actually we had an automated tool for detection of breakpoints where it built multiple trees on different sections and then automatically compared the placements of this. And if you got different placement, it concluded there was some recombination. So at some point we might bring that back.
There is actually a program called Ripples from the same author as UshER. It searches the UshER tree for long branches that tend to have a lot of reversions as part of them that would have a higher score if for part of the genome, their genotype came from some other branch. Or in other words, there's an automated way to search the tree for recombinants. In practice, repetitive types of sequence errors often trigger this recognition as well. But we have a site, rivet.ucsd.edu, where they run ripples, and somebody with infinite time on their hands, I suppose, could go through those and evaluate them.
So just related to that, often when you see amplicon dropout, there are some amplicons that drop out more than others, and there's sites that you will be very familiar with that can get flagged. Is that something that happens in ripples? Is that like, could you filter the output by something that would be more or less likely to be due to sequencing error?
Good question. That I wish Yatish were here. Often the ones I've seen trigger recombinant detection or when there are kind of like additional mutations. I don't know if it's untrimmed adapters or whatever, but sometimes 3 mutations within a few bases of each other will suddenly show up in a bunch of sequences, and that's one thing. Dropout, if they have a lot of reversions in common. However, I've been filtering things out of the tree that have a lot of reversions just to avoid massive problems caused by amplicon dropout.
Well, another issue you have is that often recombinants with similar breakpoints, independent recombinants will kind of cluster together in some non-phylogenetic way. So that also is a problem of ripples. So, I mean, there could be that we know that there are lots of recombinants and we just don't give them names because there are only a few sequences. And when something grows, it usually has a very clear demarcation as, there's a big polytomy, there's one good founder and then 50 sequences coming off that. That's for the practice, not the theory.
Great, speed round. Can I get 1 minute each from you? So how would you say different classification systems, so for example, finer grained or coarser, impact our understanding of the virus evolution or of public health responses?
I'll go first. I think there's definitely a place for both coarse-grained and fine-grained. I heard on the Slacks, occasional frustration by public health epidemiologists. For example, in the Delta era when B.1.617.2 was suddenly broken out into many AY.N lineages. They would say, how am I supposed to explain this to my boss? Last week it was B.1.617.2. This week it's AY.34. I need to explain this to people. So for them, having a coarser system like the WHO Greek letters, so they could say, boss, it's still Delta. It just has some extra mutations that were observed in some place, But for more detailed study, you need the Pango lineage with its precision. I kind
of agree. I also think there's 2 different needs. One is like, if like a really big tree with a lot of sequences in there, which is maybe useful for like public health purposes, but then for like evolutionary biologists or people who also want to study maybe a little bit more over a longer time than just a couple of years, there I think a really high quality tree with a very limited number of sequences, but extremely high quality and well supported with bootstrap support and calculated by either Bayesian method or maximum likelihood, I think is very valuable. And it's also in a way, it's, again, from like a researcher's point of view, it's easier to look at and to understand if you have like a tree with 100 sequences as opposed to 10,000 something like that. Yeah.
And at least technically, in principle, it's easy to turn a fine-grained system into a coarse-grained system simply by saying, ignore everything fine-grained below that level, which is essentially what the Nextclade clades are now. We just sort of say, that JN1.11.1 is something that people should pay attention to. and all the other Pangu lineages don't have a mapping. They just belong to something else. So that is kind of maybe this operational way that you can say that you can ignore all the fine-grained stuff if you only care about broad shifts. But here are the kind of major groupings that you should be looking at.
Wonderful. Thank you very much